Hi, I'm Shadi Darvish Shafighi, and I'm so excited to talk about CACTUS, which is about integrating clonal architecture with genomic clustering and transcriptome profiling of single tumor cells. Uh, Here in this work, we are trying to go from a tumor clone genotype to the phenotype. And for doing this, we have two types of data. First is whole exome sequencing data, and from whole exome sequencing data, we can get the evolutionary tree using existing methods. And by having this, we have clone genotypes. On the other side, we have also single cell RNA-seq data, and from this data, we can get the mutations and we can match these cells to the clones by their mutations using the probabilistic model that we designed. Also, we can get the gene expression phenotype of the tumor clones, which is so exciting. But there are some problems. First of all, we have single cell RNA-seq data and it's noisy. So the solution that we had is that we clustered the we clustered the single cells into some subgroups, and so we are using additional information of the clustering, and we are gaining some statistical power using this additional information. And all the cells inside one cluster can go to the one clone. But there are some other problems as well. First of all, you know that evolutionary tree can be imperfect, but we are correcting this evolutionary tree inside our probabilistic model. Also, the clusters of the cells can be imperfect as well, and we are correcting these clusters inside the model as well. And after this matching, we can get the gene expression phenotype of the tumor clones. But this is the overview of the CACTUS. We had two patients, two follicular lymphoma patients. First was a 37-year-old man, and second was 51-year-old woman. And for both patients, we have whole exome sequencing data, single cell RNA-seq data, and single cell B-cell receptor sequences. From whole exome sequencing data, we can get the evolutionary tree using existing methods. From single cell RNA-seq data, we can get the mutation read counts, and you can see here the rows are corresponding to the mutations and the columns are corresponding to the cells. And from the single cell B-cell receptor sequences, we can get the clusters. We use B-cell receptor sequences because of their important role in the follicular lymphoma disease, but this model can be used for any other clustering that we can say that all the cells inside one cluster can go to same clone. In these clusters, we have some clusters with only one cell, which we call singleton clusters, and we have some clusters that have more than one cell, which we call the multiplet clusters. And we expect to gain more certainty when we have multiple clusters, because in this case we are using this additional information of the cluster. Using these three types of data, we applied CACTUS, which is a probabilistic model, to infer a clonal assignment of the B-cell receptor clusters. And here you can see the assignment. The rows are corresponding to the clones and the columns are corresponding to the cells, and you can see the binary assignment here. This is the overview of the model, probabilistic model. In these kind of models, we know that all the white nodes are corresponding to the hidden variables, and the shaded nodes are corresponding to the observed variables, and the arrows are showing the causal relation between the nodes. And here I will discuss this part of the model. The hidden variables that I'm going to talk about are firstly CIK, which is the true or corrected genotype of clone K at variant position I. Also, we have TJ, which is the corrected cluster assignment of the cell J, and IQ, which is the clone assignment of cluster Q. And this is the most important variable because at the end we want to know this clone assignment of the cluster Q, any cluster. Also, we have two important observed variables here, AIJ and DIJ, which are corresponding to the alternative mutation, alternative read counts and total read counts. And for AIJ, we consider binomial distribution because it's natural that when we have total read counts and we select some of them as alternative read counts, we can consider binomial distribution for that. Of course, we have some probability as well, which is theta here. And we used Gibbs sampler for parameter estimation. Okay, here is I'm showing the Cardellino, which is published in Nature Methods, and it's the baseline of our model, and we compare ourselves to them, of course. Here's the overview of the Cardellino. They also have the whole exome sequencing, the single cell RNA sequencing, and they use the mutations for mapping the cells to the clones by their mutations to the genotype of the clones, actually. It's the CACTUS baseline, but without the additional clustering information. So, as I said, the single cell information could be noisy here. And here I will show you the results of CACTUS and Cardellino. Actually, this is the results of probability of the assignment. Firstly, in the first row, I show the evolutionary tree. And in the second row, you can see the probability assignment for the multiplet clusters, the clusters that have more than one cell inside of them. And the third row is corresponding to these clusters that have only one cell inside of them, which we call singleton. And first column is corresponding to the CACTUS, second column is corresponding to the Cardellino, and here the rows are showing the cells and the columns are showing the clones, and the values are the probabilities. So if we have more probability and the color is more intense, it means that we have more certainty in this assignment of the cells to the clones. And you can see here in the multiplet clusters that we have extra information. The certainty of the assignment in the results of CACTUS is much more than Cardellino. But in the case that we have singleton clusters, the clusters that have only one cell, so we don't have this extra information, and the results of CACTUS and Cardellino are similar, and both of them are not very good, actually. And it's like that we are not that much certain in assignment of the cells to the clusters. Which can cause by the number of reads, the coverage, and other problems of the data. We have also another data, 1218. You can see the tree and the results of CACTUS and Cardellino here. And you can see here, even when we have singleton clusters, we are more certain about the assignment. And both Cardellino and CACTUS are certain and same in singleton and in singleton, actually, but in multiplet again we are more certain than Cardellino here. And we also calculated, somehow measured this certainty by calculating the average entropy and gene index. Here in this table, you can see we both the numbers that are better, and you can see that CACTUS were better in all the cases, but the difference between CACTUS and Cardellino in multiplet clusters that we have more information is much bigger, and you can see that CACTUS was much better. Also, we did some validation by the gene expression data. Actually, we use the gene expression vectors and we reduce the dimensionality of this gene expression to the two dimension using UMAP. And here you can see the left hand side are results of the CACTUS and right hand side are results of Cardellino. And this first row is the results corresponding to the multiplet clusters, and the second row is corresponding to the singleton clusters. And each point here is presenting one single cell. And we expect to see that the cells with same color, which means the same the cells from same clone, actually be in the same position and make a cluster themselves. And you can see in here in the multiplet version, CACTUS, the results of CACTUS can show this clustering. You can see some green points here and pink points here, yellow points here, and you can see the clusters of the cells. But here in the Cardellino, all the cells are together. Also in the singleton version, CACTUS and Cardellino, both of them, all the cells are together. And we also did this validation for the other subject, 12118, and we kind of see the same results over here as well. And then we also compared our assignment together. We see the assignment of CACTUS and assignment of Cardellino, and we wanted to see if we agree or not. And here you can see the columns are corresponding to the clones and the rows are corresponding to the clusters. And the number that you can see here are the number of cells inside one cluster. So, for example, cluster B have 131 cells inside of it. And you can see the two subjects and the results of CACTUS and Cardellino. And in the subject 144, if you remember, the certainty of the assignment wasn't very good. Specifically for Cardellino, it wasn't very good, and you can see the agreement is not very well. But in subject 118, you can see the agreement is almost very well, and we assign the cluster, the cells inside the clusters to the same clones. In summary, I can say that CACTUS is matching single tumor cells to their clones of origin by common mutations, and it benefits from the additional information of contained in clusters of single cells. And here we can use any other clustering. Actually, in this project, we use B-cell receptor sequences because they were related to the disease that we were studying, but you can use any other clustering that you can say all the cells inside one cluster can go to one clone. And at the end, I can say that CACTUS is a step forward in resolving the impact of clone genotype on their functional diversification. And at the end, I wanted to thank my supervisor, Dr. Eva Sturek, and our great collaborators in Leiden University, Shimon and Kes. And thank you for listening. I'll be happy to take any questions.
Thank you very much for the interesting talk. So we have time for some question in the room or question from the Q&A. We'll just check if we have some. So we have no question from the Q&A. So I will have a very quick question. So you are considering additional information for your mapping of tumor cells to their clones. Are you thinking about adding over additional features to improve your tool?
For improving, we have some stuff in our mind. For example, we can do we can infer the evolutionary tree inside our model, or we can infer the clustering inside our model, because now we are doing the clustering outside the model and we are just collecting the clusters inside the model. But we can do it inside our model, and we are working on that actually for another project.
Okay.
Uh,
Okay, thank you. And so you think this will improve the result actually of the
yes,
mapping?
yes, actually, it can improve the results. This inference, because you will reduce the noise of other methods or other clustering methods. So,
Okay. Thank you very much. And
thank
we
you.
can thank again. Sorry, there
Thank
is a
you.
question in the Q&A. Okay. Okay, just a question just popped up. Thank you from
Okay.
Anna Isabel. So the question is, do you see CACTUS applicability to study primary and met compartments?
Uh, um, Actually, I don't know. Primary, I don't know the area.
To study primary and met compartments.
Actually, I don't know about that.